Bioinformatics is a very wide area. In this presentation, I will focus on some topics that I think are necessary for a biologist or a molecular biologist to know in order to make use of the vast quantities of data now available online. If I were to sum up the content of this presentation, I could do it like this. I will start with challenges of bioinformatics, sequence representations. How do we represent biological sequences such as RNA and DNA in bioinformatics? The FASTA and GenBank formats that are formalized ways to write or store biological sequences. Especially important for the latter format is the feature table, where most of the interesting information is stored. I will talk about some biological sequence databases, the application of cryptographic hash functions and what they are, sequence alignment, that is comparing similarities between sequences, and then how do we quantify mutations, how do we find sequences that are almost the same. If we take a step back and look at what we might think is common for everything that we call bioinformatics, we can say that it's an interdisciplinary field where we develop methods to store and retrieve information, organize it and analyze it. We have now lots and lots of biological data, and a big part of the reason for this is the development of omics technologies. You can see here metabolomics, genomics. These omics technologies tend to generate large amounts of data. For instance, metabolomics is all the metabolites in the cell, genomics is all the genes, proteomics all the proteins, transcriptomics all the mRNAs. And this is necessarily, since we have thousands or tens of thousands of genes and molecules, this necessarily becomes lots of information to store and lots of information to interpret. For this reason, we need bioinformatics, which is the application of computer science in biology. I like to show this slide as an example of how much information is really in our genome. If we assume that we print our genome, the A, Cs and Gs and Ts of our DNA in books like this one. It's a very large book. You can compare here to a soda can. We need 42 of these books in order to print our own genome, the human genome. As a comparison, we might need 200 pages for E. coli, which is an important laboratory organism. We need 2 of these books for Drosophila, the fruit fly, which is an important model organism. And in order to write the genome of this organism here, which is the marbled lungfish, Protopterus atopicus, we might need 40 times as many books as for the human genome. Just to say that we're not on top of the evolutionary tree, at least not in this respect. To organize biological data, we have computers. These computers have developed at a pace that is difficult to understand, really. In the mid-1980s, a typical supercomputer, such as this one, the Cray XMP, would cost $15 million, whereas a computer of equal capacity today, or now several years ago, is the size of a credit card and costs $35. This development is sometimes called Moore's law, and it means that computers double their capacity every 2 years, and they cost half as much. If we plot Moore's law in a logarithmic diagram, we get this straight line here, which is then how much computers get more efficient and cheaper with time. If we compare this with the development of the price of DNA sequencing, the new technologies have really lowered the cost of DNA sequencing in a way that is much faster, in fact, than the cost of computers. So this particular diagram ends at 2012, and since then sequencing has become even more available economically. Classical genetics, such as the genetics of Mendel here, is really an example of forward genetics. We have a organism of some kind, and we try to isolate mutations, such as this one, and then from the mutation we try to map out which gene is really responsible for this phenotype. The availability of sequence information, and especially genome sequences, facilitates something called reverse genetics. And we really do the opposite. We start with the gene sequence. We guess where the genes are located on the genome. We introduce specific mutations. That is, we delete the gene or we change the gene somehow. And then we see if this has a phenotype and which one it has. This is really easier and faster than this strategy, and we can more easily cover all the genes in this way. Next, I will talk about sequence representation in bioinformatics. That is, how do we actually store the information about biological molecules, especially sequences, in the computer. Most programming languages used to program computers have a concept called string, that is, this kind of data type or variable that is used to store text on a computer, which is very often what we use computers for. And in bioinformatics, we use this data type to store DNA simply as strings of A, G, C, and T on the computer. So we simplify this molecule here into a piece of text or a string with just A's, C's, G's, and T's. This is, of course, a very large simplification, and we should always remember that this is a representation of DNA that is very simplified. Because of this simplification, we need to enforce some conventions when we use strings to describe DNA. One of these conventions, maybe the most important one, is that we always write DNA 5' to 3'. So if we have a molecule like this that we want to describe in a computer, we would write this C, T, A, G. If we don't use this convention, if we write the complement of a DNA molecule or something like that, we need to indicate this. If we just consider a string containing A's, G's, and T's, we always assume that it is DNA 5' to 3'. This simple representation also doesn't allow to tell much about the nature of the molecule itself. We have this string here representing DNA. This could be single-stranded DNA, like the example here, A, T, G, C, 5' to 3'. It could also be a double-stranded linear molecule, like this one. We normally assume that we don't record the complementary strand normally in bioinformatics. And it could also be a circular sequence like this one. So in case the DNA here represents a double-stranded sequence, there is an equally valid representation, that is this reverse complement strand here. One of these representations is not more correct than the other. There is this simply something we need to store somewhere else, the fact that this molecule means single-stranded, double-stranded, or a circular DNA molecule. This here is a circular double-stranded molecule, and there are many more representations of this molecule than for any of the others. A circular double-stranded DNA molecule is a molecule like this one, where the first, so to speak, nucleotide here is connected to the last one in the end. And when we assume, for instance, when we want to represent this kind of molecule by a simple string, we assume that this nucleotide in the beginning here is actually the one we see here at 12 o'clock in our circular molecule. And the last one is actually the one just before 12 o'clock here. This means that we can arbitrarily choose where this 12 o'clock is. So we have as many representations of a single strand or double-stranded DNA molecule that is circular as we have nucleotides in the sequence. So in this case, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. We have 14 different representations of these. This is an exercise in figuring out an algorithm for bioinformatics. We assume that we have these three sequences. We think that two of the sequences actually represent the same double-stranded DNA molecule, circular that is, and the third one is not. We have the sequences here. It's not entirely obvious which two sequences are the same. Maybe we can guess that this beginning of the sequence here, CTGAT, seems to be present here in this sequence, CTGAT, and then it appears very similar. But finding that out for a larger sequence is very difficult to do manually. A solution to this could be to check for two conditions that have to be fulfilled. First, the lengths of the sequences has to be the same. And if you look at the sequences in the previous slide, they are all the same length. So this is true for all of them. The second is that if we add one sequence to itself, we should be able to find the other sequence inside of that sequence as a subsequence. And we can choose, we have to compare this 2 by 2. For instance, if we have sequence 1, we can take sequence 1 plus sequence 1, and we should find sequence 2 inside. If you don't understand this, think a little bit about it, and I think it will be clear. You can actually test this on your computer without using any kind of DNA editor or special software. If you open up your Microsoft Word and then you take this sequence, for instance, sequence 2, and add the sequence to each other twice, and then you take the sequence 1 and then you make a find inside the document, you will find this sequence inside the 2 plus 2 sequence. And this means that these two sequences are representations of the same circular DNA molecule. You should try this if you can. In order to safely store and retrieve our sequences from our own computer or from a database online, we need to follow a formalized format for our sequences. This is also important for other software, other bioinformatics software, to understand our sequences. One of the simplest formats that we use is the FASTA format. It contains simply of a line, first line, which is this one, the header line. And the first part is a more than symbol, and then a label or an identifier, we see in the literature label, identifier, or name for this yellow part. We have two examples here. This is a protein sequence and this is a DNA sequence. If we add a space, no spaces are allowed in the label, by the way. We separate with a space here, we can add more information about our sequence. This here is, as I said, a protein sequence, and this is a DNA sequence. Both of these allow spaces within the sequence, for example, to facilitate reading. But we cannot have empty lines within this sequence. We can also not have empty lines between the header line and the first line of the sequence. Based on what you learned in the previous slide, which ones of these 11 sequences are correctly formatted FASTA sequences? If we start with one, clearly this is not valid because we have to assume that this is the sequence information, and we need to have the identifier and the sequence on separate lines. This one here has a wrong symbol in the beginning, so it's not valid. This sequence here is valid. We have a P here, which we don't expect in a DNA sequence, but we'll get back to that. This sequence is valid. This one is valid as well. This is normal DNA. This one is valid. This is RNA. This sequence here is also valid. It's a protein sequence. This one is not valid for the same reason as sequence 1. This is okay. We have spaces between the characters. This is also okay. It doesn't matter whether we have lower caps or caps in the sequence. It doesn't matter for the interpretation at all. This is also okay. We have some spaces here. This is not okay. We cannot have an empty line between the identifier and the sequence information. If we go back to sequence 3, what is the problem here? The problem is that this could be a protein sequence. All of these identifiers here are valid for some of the codifications of protein sequences. But it could also be somehow an error in the DNA sequence. We don't really know that. And this is maybe a drawback for the FASTA format. We only have one line of sequence information, and this might not be enough to store all the meta information, the information around the sequence that we are interested in order to know, to protect the information. These two tables here represent common codes used in nucleotides and in amino acids. And you can see that we cannot really tell apart very easily whether a sequence is actually a protein or a DNA sequence, for example. We have ACs and Gs and Ts here, but we also have many letters that represent dubious codes in the sequence. We have the same kind of letters here in the amino acid code. That means that it's better if we save some kind of information in the sequence itself that tells us basic things about what we are looking at, if it's a protein or a DNA and many other things. There are several sequence formats that are better than the FASTA format in order to save information that is important for the sequence other than the sequence itself. One of the most important of those formats is the gene bank format, which is, like the name implies, the format that GeneBank uses internally. This format has 3 parts. The easiest to tell apart is the sequence itself here below. This can be DNA or protein, just like before. And then it has a header, which is this blue text here. The header is important. It has several identifiers. One of the most important is the locus, but also the accession. And I will go through why accession is important. You can see here the size of the molecule. This is important because you might lose some of this information if the sequence is poorly copied, for instance. We have a date, we have what kind of molecule it is, DNA in this case. We have the topology, which is linear in this case. So we know a lot of this, what the sequence should be. You can also sometimes see dsDNA to indicate that it's double-stranded DNA. Sometimes this is missing. It doesn't really mean that much. This molecule is, for instance, double-stranded. The definition here is simply a short description of what this is. So this is a gene of the cytochrome C for yeast. And then we have other kinds of information like the source, which organism it is, where does this organism fit into the tree of life, references, where was this sequenced, who sequenced it, who published it, where can we read more about it. And then maybe the most important part of the sequence is this feature table. This sequence in the text in green here. This is where we can we can distinguish different parts of the sequence. For instance, CDS here, it means coding sequence. And this sequence here contains an open reading frame, a coding sequence, something that codes for the cytochrome C that we see there above. So between 249 and 578, that means 249, it's around here, ATG, until 578, that is here, around here. We have the gene. This is extremely useful. We can have many of these features. It's in the form of a key value list in a way. These are the keys, various source, CDS. The source is all of the sequence from 1 to 857, and within there's this part that is a coding sequence. These identifiers here that starts with a slash are information, pieces of information that belong to this key here, and they can tell us more about what this key is actually about. We have an unnamed protein product, a note, that is where a codon start and a protein translation and an identification of the protein itself. If we go back to the header part of the sequence, the first blue part, again, the most important part, the most important pieces of information are in the first line, that is the size, what it is, the topology. And the second most important might be the accession. And why is this? The accession number is the key to this sequence in GenBank, so that this number here, or this code, is uniquely describes this particular sequence file. Why is this accession number important? They are important because they are stable identifiers of any sequence in GenBank. So, for instance, sometimes you maybe discover an error in a sequence somewhere, like this example here, the PUC19 vector. It has two versions. So the sequence, the accession number is L09137, and the first sequence file that was published had this sequence number, and then the version 0.1 here in the next line. Many years later, or in this case the year after, an error was discovered, and they made a replacement sequence for this plasmid, and you located here a 0.2 to indicate the version 2. But this original sequence is still there, so if you somehow refer to a sequence by the accession number, this will always be there in this shape. They will never change information once they put information in GeneBank. This is very useful because you don't have to actually store DNA information on your own computer. You can simply store this accession number to save space and work for yourself. Then go back to the feature table, which I consider the most important part of the GeneBank sequence, because here the most useful information tend to be stored. In this sequence, we only have two features, two keys in the feature table. But there are many examples of GeneBank files that represent an entire genome, and we have thousands of these keys in the feature table that, for instance, represent every gene in the genome of that organism. Again, just to repeat, the key and the coordinates within the sequence where we can find the feature, in this case the CDS, the gene for the cytochrome C gene, so between 0.249 and 578. These lines here that belong to this feature are called qualifiers. They start with a slash, so slash note means some kind of information, and then these are free to you can freely invent new feature qualifiers, but it's best to use feature qualifiers that are accepted in order to make your sequence more useful. If we look further into the key value pairs of the feature table, we can actually also invent these keys as well. We are completely free to invent keys for the table, but again, it's best to use keys that are already accepted in biology or bioinformatics. If we look at this key again, we might read this as, or this feature. It's a CDS coding sequence beginning at 23, ending at 400, with a product called alcohol dehydrogenase, and the gene is called ADH1. And these two are qualifiers, as I just stated. They are valuable to tell you something more about this feature. The keys, this is the CDS examples, but there are more. And these are found in sequences on GeneBank. The first is the miscellaneous feature. This could be anything at all, but you want to say something about that part of the sequence. The CDS, the protein coding, ribosome binding site, RBS, origin of replication, where, for instance, a plasmid or a genome could start replicating. Protein binding area is where, for instance, a transcription factor might bind on DNA. tRNA could be a transfer RNA, gene encoding a transfer RNA. And a primer bind is where a certain primer binds on the DNA. This is something that we add our own primers, and if we do, we use this feature table key. The feature qualifiers always take this format. You have the qualifier, and then an equal to, and then some text. This text has to be enclosed in citation marks here, and it can be anything at all. Anything we wish to write here is valid. Location of a feature doesn't necessarily have only a starting point and an end point. In this example here, we have two locations from 544 to 589, and then 688 to 1032. And there's roughly 100 nucleotides between here. And then we have the keyword join. So this we might read that this coding sequence consists of this part of our sequence file and together with that part. So we should put them two together. This might be due to, for example, a intron. In the case of eukaryotic genes, we might have an intron here in between that is spliced out of the coding mRNA at a later stage. Now we would like to cross the actual sequence that we have with an example feature. This file here represents perhaps the smallest and simplest gene bank file possible. We have here a miscellaneous feature starting at 10 and ending at 14. And if we look at the sequence, this 1 indicates that the first nucleotide is actually nucleotide 1. So the index starts with 1. And we have the 10th nucleotide here, T, and then ACTA, and this A is the 14th nucleotide of the sequence. I've made a copy of the molecule here. This is 5' to 3'. This is a complementary sequence 5' to 3' here. And I have numbered the nucleotides so that we can actually confirm that this is the 10th to the 14th nucleotide. This blue text here is actually the exact nucleotides for which this feature is specific. This isn't always so, and I will come to that in a moment. But if we are asked for the sequence of this specific feature, it is TACTA and nothing else. If we take a look at this sequence file, it is almost as simple as the one in the previous slide, only our location here has the keyword complement. And this means that the feature is between 10 and 14, just like in the previous example, which means between this nucleotide and this nucleotide, the sequence is exactly the same. But the complement here indicates that the sequence feature is actually on the complementary strand. So if we look at our double-stranded model here, the sequence, our actual feature is this one. It's the same sequence but on the complementary strand. So if we would be asked for the sequence of this feature, it is actually TAGTA, because this is the sequence of this part here, this blue part here. And since we have 5' here and 3' here, we have to invert this sequence, so it's TAGTA. Now let's look at almost the same example, but now with a split feature. We have positions or locations between 6 and 9, 13 and 16. and these are to be joined. And we can identify the location here between 6. This is 1, 2, 3, 4, 5, 6, 7, 8, 9, and so on. And I've done the same kind of model here. If we are now asked to give the sequence of this feature, it is GATC, this first part, and TACT, the second part here. And now for the final and most complicated example here. You can actually combine these two keywords here, complement and join. And in this case, this feature here consists of positions 6 to 9, 13 to 16, just like in the previous example, but the complement. So our sequence is actually on the complement strand. And how do we solve this? We actually have the same part of the sequence as in the previous example. This is exactly the same, but the sequence we want is on the complement strand. So in this case it's AGTA, GATC, like this. So the final sequence in this example is AGT, GATC. Since we have 5' here and 3' here of the complement strand. As we saw in the previous slide, it's quite a lot of work to manually extract the sequence of features manually from the GenBank file. Luckily, we have a very nice tool to do that for us, and that is our ape plasmid editor that you can get here if you don't already have it. If you open the sequence as a GenBank file, that is, the sequence that you open has this format, it will be automatically recognized by ape, and the feature will be colored in the sequence itself. You can also copy paste this sequence. If you copy all of the sequence, it will also be recognized as a GenBank file by ape, and you will get this coloring. It's a very nice tool to visualize features. On a practical note, if you have a sequence in one format and you want the sequence in another format, there are several services that you can find online in order to make this into conversion automatically. If you Google readsec like this, or secret spelled like this, you will find two of these tools at least. This is one of the readsec services that I found, and this is the emboss. This is the European EMBL, basically EMBL service. And you can use this, you just paste your sequence in the window and you say which version, which sequence format you want, and possibly which sequence format you have, and then you just click a button and it's automatic. Let's now shift a little bit to think about all the kinds of biological sequences that are available to us nowadays. There are two kinds of databases online that are usually free for everyone to browse. There are primary sequence databases and there are secondary databases, or gene-centric databases. And these primary databases, they contain experimental data, sometimes raw data, such as nucleotide sequences, protein sequences, and structures. Examples of these are GenBank, which we have used, the American Biotechnology Information Database, the European Molecular Biology Laboratory Nucleotide Database, the MBL, and the DNA Data Bank of Japan. These three databases contain more or less the same information, although organized slightly differently. Then there are secondary databases, or gene-centric databases. These contain data derived from analysis or treatment of primary data. So all the data that we can find here, we can also find in the primary databases. But we can usually also get some more information around the sequences. Usually these are organized around a specific organism or a specific type of protein or maybe a disease or something like that. Examples are Flybase, which is the Drosophila genome database, Saccharomyces genome database, and the human genome database. These databases organize around the specific organism, and we can know much more about each sequence if we use these databases. I would like to use this example to illustrate the differences between a primary database such as GenBank and a secondary as the Saccharomyces genome database. Suppose that we would like to know more about hexokinase of Saccharomyces cerevisiae. So this is an important enzyme in this organism. It catalyzes the phosphorylation of glucose in the first step, one of the first steps of glycolysis. So where do we start looking for that? We have these two options, and we shall compare them in the slide to come. If we start using the GenBank and we simply put in the organism name and the name of the gene or the protein we want to know about, we get lots and lots of results. We get here we get 3,600 results roughly. Probably get more nowadays. This is an old slide. And it's not immediately clear which one of these are valid results. These are probably not interesting, any of them, because they belong to different species here. And we simply get them as results here because they contain genes that are probably similar to the hexokinase. We can see here we have more valid links here for hexokinase in the gene database of GenBank, but it's still a bit difficult to interpret these results. If we, on the other hand, do the same search at the Saccharomyces genome database, we immediately get the valid results here, or the useful results. We get to know that there are two hexokinases, isoenzymes, in this organism, and there is a third gene that is possibly also a hexokinase. And we get all this information because the database contains so much more information regarding Saccharomyces cerevisiae. So the take-home message here is that if you know that you're interested in a specific organism, it's better to use one of these secondary databases than GenBank, because you get to your result much faster. Now I will move to talk to you about cryptographic checksums. This is a technology that is not specific for biology or bioinformatics, but it's a kind of technology that we use every day to enforce security and to verify integrity of data. It isn't central to bioinformatics, but I use these tools. I use these checksums as a teaching tool in this course. So I would like to explain to you a little bit about what they are. It's not necessary to understand them in detail. The mathematics of it might be difficult, and it's also not important. It's only necessary to understand how they work. If we look at some sequences in GenBank, sometimes there is this strange code here attached to the sequence, and this is a checksum that is called CRC64. And what is this? This is simply a sum, a small short sequence that is calculated based on the content of the sequence. We can recognize that this is a protein sequence. And we essentially store the sequence information plus this code that depend on the information for security reasons. Imagine if this sequence is edited by mistake, so that we delete one amino acid or we replace it with something else, then this checksum will not be valid anymore, and we can detect the error. So these cryptographic checksums are hash functions. We can use these terms interchangeably. And this is basically this process. We have a piece of information. We can use an algorithm to calculate a checksum. And this is a relatively small piece of information here. And because of the way this is done, it's impossible to go back. We cannot use the checksum to identify the piece of information directly. We can only use the information to calculate the checksum. So it's deterministic. We have some properties. That means that one piece of information always gives the same checksum. It's not possible to go back. This is infeasible, second point here. And a small information change. We can have a very big amount of information here, and we change a small part of it. This checksum should be very different. And lastly, we have two pieces of information that are different. They should never get the same checksum. So as I said before, checksums or cryptographic checksums are used in many places in society, and I will show you a specific example here, amusing way you can actually use these checksums. If we imagine that an inventor has a very nice idea, and then he writes this idea down, and this inventor has a computer, so he writes on his computer, but he doesn't want to publish yet. He has other work to do, or he wants to complete his ideas. So what does he do? He doesn't want to publish, but he wants to say that he was the first person to invent this, or to have an idea, to have this idea. What he does is he takes the completed description of his idea and he calculates the checksum, for instance this, and then he publishes this checksum in a newspaper. This is actually a real example. We see these hash function codes here printed in the newspaper. Nobody knows on the newspaper what these mean. They only mean something to the person who made them. And then he can complete his studies, or make additional observations, or something. And then he can take his new, maybe updated piece of information and publish. And he can say that, well, I might have published at this date, but I already had this idea at this time when this newspaper was published. So if somebody has the same idea in this time here, he can say that he had it first. This may seem a convoluted way, but this has actually happened in reality, and I will give you a nice example. If we go back many hundreds of years, the famous Italian astronomer Galileo Galilei observed Saturn for the first time using his telescope. And what he saw was these shapes. We now know that Saturn has rings, but he couldn't really see that on his using his telescope. He thought that these were moons, or that there were three parts of the star. He thought that Saturn was a star. And he observed this, and he realized that this was a significant observation, and he wanted to tell the world that he saw it first. So he had a problem because he made observations so fast in this time that he didn't have time to publish it. And he had to find a way to gather his data, but still make sure that nobody would dispute that he made his observations first. So what did Galileo do? He wrote down this sentence in Latin, and we can translate this sentence into this text in English. I have observed the highest of the planets, Saturn, three-formed. So this sentence means that it sort of describes his observation. The highest of the planet, the Saturn, what was the planet furthest away that could be observed at the time. And three-formed is then the description of his observation. And then he took this sentence here and he made an anagram, so he scrambled the letters into this. And then he mailed this code to many of his co-workers, co-scientists at the time, like Johannes Kepler in Poland. And none of them understood what this meant, but he knew that if they had this information, he could later prove to them what this anagram. They tried to solve it, by the way, and they were not successful. But he could later then say that since I mailed you this anagram, I can now show you that it means this, and I made this observation before you. We also normally use one of these checksum algorithms during the test and the bioinformatics test in order to ensure that the student can email safely the test several times if necessary. So this is how it works. The student gets a test file that is a computer file, and he adds his responses here in the file, and then uses a software to calculate the checksum, which is this one here, and then he emails me this file. And this is all nice and well. We don't need a checksum to do this. But imagine sometimes the email doesn't arrive for whatever reason. Then the student might want to send it again. The problem is only that if this happens after the test is finished, then I cannot be sure that the test wasn't changed. But what the student does during the exam, he writes this checksum on a piece of paper and hands it in. And when the test file arrives, I also calculate the same checksum. And these two, if these two match, I know that this test file is exactly the test file that was handed in at the time of the test. And this makes it possible to email the test file very much after the test, because I can always verify that it's the same version. We also use the SegWid checksum for DNA sequences as a teaching tool. We have this piece of software here that calculates 3 checksums: the SegWid, the L-SegWid, and the C-SegWid. And if we calculate this checksum for this sequence, in this case it's 30 characters here. You can see here there is no other sequence that gives this checksum, no other DNA sequence. So in this case I can put hints in the documents, in the TP documents, so that you can know if you can check your own results. Usually I give you the first 4 characters of any checksum that is asked for. The C seguid is a checksum for circular sequences. That means that it will give you the same code, the same checksum for any sequence that represents the same circular sequence. If you can see here, we assume that this sequence here represents a circular sequence. There are 5 different representations of this sequence, and there are 5 equal different representations of the reverse complement of the sequence. This means that if we would calculate the checksum of these, we would get 10 different seguid checksums. But what the software does is that it lists all of these different representations, and it picks the smallest one. If we look at all of these and we sort them, we see that this one starts with an A, T, C, C, G. This one here also starts with A, T, C, G, G, but this one is the smallest, the lexicographically smallest. That is, if we sort this list here in alphabetical order, this one will come out on top. And then we pick this one and we calculate the seguid. And this is defined as the C seguid for a sequence. We can see that here for A, T, C, C, G, the C seguid and the seguid is the same, which we would expect. The last part of this presentation is about sequence alignment. That is, how do we actually compare DNA sequences or protein sequences that are not identical but slightly different? This is a big issue in bioinformatics that deals with biological sequences, because usually the interesting part is in the differences and not similarities. And we need to be able to find the differences, we need to be able to measure them and to give them a value, a quantitative value between differences of sequences. And there are several different algorithms to do this, and I will show you a very simple application that you can actually do on your own computer. First, before we start, we should get some nomenclature right. We have often heard about homology, that sequences are homologous. This is sometimes a term that is abused when we in fact mean similarity. 2 genes are homologous if they share a common ancestor, and we need to know about the evolutionary history of the 2 sequences. Similarity is simply what we see, what we observe. We measure the similarity between the 2 sequences. This doesn't necessarily imply that they share an evolutionary history, but we should nevertheless keep these 2 concepts apart. Of course, a high level of similarity is a strong argument for homology, but they are not the same thing. In order to compare DNA or protein sequences, we do something called an alignment. And this is simply a way to group the letters of the sequence so that we have as many of them similar or many of them identical like this. So here it's obvious, for instance, that if we assume that these 3 organisms share a common ancestor at some point, we can assume that the similarities are more than the differences, and we can align these letters here with some degree of certainty. We have, of course, differences. We expect that. Some are substitutions, that is, one letter is substituted for another. And we also have deletions or insertions. We group them together and call them indels, because we don't know in this case whether this T here in the fish, if this was inserted or if it was lost in the bird and in the dog, in this case, compared to the ancestral sequence. We could, of course, look at the ancestral sequence and tell, but usually we don't have the DNA sequences. This is a trilobite here. This is probably not true. Trilobites didn't give rise to fish, birds and dogs, but it's just an example. We don't have this DNA. These are fossilized since hundreds of millions of years, so we don't actually know. So we can't say if this was a... We would say that this is actually a deletion, because in the original sequence there was an A, but we don't know that. And what we need is then a nice and efficient algorithm to make these alignments. And the alignment is such that we expect as few of these holes in the sequence. We want to have as many similarities between the words or between the text as possible. These are three alignments, three of the best alignments between these three words as an example. And the trick is here to, how do we do this efficiently? How do we group the sequences efficiently? There are two kinds of alignments that we can do. The global alignment, which is exemplified here, and a local alignment, which is here exemplified. And these are two different goals, really. In the global alignment, we take the entire sequences, the two sequences, for instance, in this case. We try to align them as well as we can. In this case, these two are quite different, so they only align here in the middle and in the ends. A local alignment, on the other hand, tries to find the subsequences that align more efficiently. This can be more efficient for sequences that are very different, because we get the similarities here and we ignore the rest. This is a local alignment because this is a localized part of both sequences. Whether this is global because globally both sequences take part in the alignment. The question is, what kind of algorithm do we use to compare sequences? How do we align two sequences or at the same time measure the similarity? Something called a dot plot is actually the simplest algorithm we can use. We don't even need a computer really for this. If we assume that we have two sequences, two protein sequences X and Y here, and they have these amino acids. What I did was I plotted here the X sequence, we can compare, it's the same here, and the Y sequence here in the Y-axis. And then I filled in these squares, this collection of squares here, and whenever I have the same amino acid in X as in Y, I put a cross. You can see here we have an A, E here, Y, another Y here, so another L here, and so on. This is very simple to do. You can in principle do this manually. I didn't do that. I have a Excel sheet that does that for me, and you can find it in among your teaching files, course files. It's called pairwise alignment examples. And if we just look at this picture, we can immediately see that there's something going on here. We see that there is the diagonal in the dot plot. And if we look at that more carefully, we see that this diagonal represents a sequence that is exactly the same, a subsequence AESLMG. No, sorry, AESLM. AESLM. We can easily see that this is a quite substantial part of the two sequences. substantial because these are short sequences. And it's not really easy to see that. AESLM here and here, AESLM. But if we plot it like this, it's very easy to spot. And this is still used with the automatic calculations of this matrix here, because it's very simple algorithm, and it's very easy to interpret. So how do we turn this very simple algorithm into something that a computer can understand and interpret? We could, of course, create this kind of matrix for every sequence pair that we want to analyze, and then look for these runs of diagonals here to see. But it's unfeasible. We have so much sequence information that we actually need the computer to do this for us. And we can easily do that by modifying our original algorithm. So instead of putting an X in these cells here in the matrix, we put a number. And we put a number 1 for amino acids that are the same here, and we put not only the number 1, and we put the number 0. We don't see the zeros here in this matrix, but whenever we have different amino acids, we put a 0, and we don't see them here for clarity. But we also look at the upper left cell every time we do the calculation, and we add whatever number is there. So in this description here, we are calculating this cell here, and we add whatever number that is in this cell. So for instance here, we have E and E, which gives us 1, but we have 1 also in the upper left cell here, so the sum is 2. So if we do this for every cell in the matrix, when we come to this run of different sequences that is the same as in the previous example, we add 1 each time here. Every time we pass these cells, it's 5 here because it was 4 here, and we have another here. And this makes it very simple afterwards to ask the computer, where is the longest diagonal, where is the longest shared sequence between X and Y here? Because we can simply ask the computer, what is the biggest number in this matrix? And this is kind of a question that computers are really good at. We find that it is 5, there is no other value here, and then we simply trace back here until we reach 0, and this is our longest diagonal. So with this simple modification, we have turned this algorithm into a computer algorithm that can be automated to find shared sequences between any two sequences. Test our new algorithm with the different sequences. Imagine if we have the exact same sequence in X and Y. What do we expect? We expect, of course, this diagonal here that is the same sum in the end as the length of the protein sequence, because according to the algorithm I showed you, you calculate this and we always have the same identity here in the diagonal, E versus E. We have 3 in this box, so it's 4, and so on. We also have some sequences here, that short ones that are the same. They're internal repeats. AE and AE are shown up here in the diagonal, but also once more here and once more there in the other part. Well, let's test our algorithm some more. What happens if we compare two sequences that are very much the same, but they have one internal difference here? As you can see, they're almost exactly the same. The only difference here is that N in the upper sequence is an E in the lower sequence. Other than that, they're exactly the same. Let's put the two sequences here, the N sequence, the X here, and we can see that our algorithm gives us the sum of 10, which we expect, but nothing here because it's 0. And then we start all over again and we get the sum of 10 again, because this sequence here is 10 and 10 is 10 amino acids, and this one as well. So we can conclude that our algorithm is local alignment, and we can somehow say that it fails to appreciate the true similarity between these two sequences. They are quite similar, so a global alignment procedure would have been more appropriate here, perhaps. But the way we see, the way we expressed our algorithm, we don't reach this conclusion. We get two runs of similarity that are 10, and what we would want to is some kind of algorithm that shows us the true similarity between these two sequences, and we can perhaps modify this algorithm to also deal with a global alignment. We can in fact make a very simple modification to our algorithm to enable to make it able to cope with this kind of situation. If we simply modify the rules slightly so that we not only add the upper left value when we have a score, we also do it when we don't have one. So in this case, the match score is 0 or 1, but we nevertheless add whatever it is in the upper left cell. So when we arrive here at 10, what happens is that next time we come to this cell, we have a 0 because N and E don't match, but we have 10 here in the upper left corner, so we simply print 10 here, and then we continue, and we arrive at the final value here of 20, which is somehow more indicative of the true similarity between these two sequences. This is somehow a semi-global alignment algorithm at this point. Let's test the new algorithm again, this time with indel difference between the two sequences. Here, instead of switching the E for an N, I deleted the E here in the Y sequence. So this is a different kind of difference between the two sequences than the ones we have seen so far. And we can see here that our simple algorithm fails here because we arrive at 10, and then we have 10 here in the upper left corner, but we have a different amino acid because here there is this shift in the sequence, and we just continue all the way down, never arriving to any other value than 10 because the differences are all the same. But on the side here, we see that we have again the similarities, but shifted a little bit to the right because of this deletion in the Y sequence. So we get 1 T and 1 T here. We get 1, 2, 3, 4, 5, 6, 7, and we get 10. And this is somehow a result that's not really very good, because we fail again to appreciate the difference or the true similarity between these two sequences. We get a sum of 10 here and a sum of 10 here, but it doesn't really capture this fact that the two sequences are quite similar. So the question is, can we again modify our algorithm to be able to deal with this situation? And the answer is, of course, we can. What we fundamentally have to do to our simple algorithm is, in order to achieve a true algorithm that can deal with both substitutions and indels, is not only do we have to take care of or to regard whatever is written in this cell here at the upper left, we also have to take into account the cell to the left and the cell above. And we should regard these values in different ways. Before, we simply took this value, xy, plus the score, that is 0 or 1 in our example. If we have a similarity, we have 1. If we don't, we have a 0. And then we add this. Now we have to also see whatever is in this cell and in this cell. And we have to invent something called gap penalty. That is, how much does it cost us to open a gap in one of the sequence? That is, instead of aligning as we did before, we need to open a space in one of the sequence and continue and this has to be assigned a cost, because otherwise we would not arrive at a proper result. So the gap penalty, we can see that as a score, sort of negative score. The gap penalty is usually negative. So what we do is, instead of simply taking this score plus whatever is written here, we compare the three values here. What is this one plus the score, the first line here, and what is whatever is written here plus the gap penalty, usually negative, and what is written here plus the gap penalty. And then we look at all these three values and we pick the one that is largest, whichever of these three is the largest, and we put that value here. And we do this for all the cells. So now we have quite a bit of calculations. For every cell that we want to calculate, we need to calculate three values and pick the largest. So let's now test our new algorithm on an example where we have an indel. And most of the time, most all over the matrix, the result is exactly the same. But when we arrive here to this cell, something happens, because we don't just look at what is in the blue cell here up on the left upper cell of the cell we are calculating. We also look at the sides here, the yellow cell to the left, the red cell above. If we take this example a look more carefully, what do we actually put in the pink cell here? I copied this here, the content here. What we actually have here is 0 and 0. We don't show that in the matrix for clarity, but we follow our rules. What do we put here? We have 0 here plus 1. 0 plus 1, that is 1, okay. We have 0 here in the upper left plus 1. We have the 1 that comes from the match score here, t plus t. And then we finally have 10, which is on the left, plus the match score here. So we put 11 in this cell 11, and then following from this, when we do the next calculation here, we see that the biggest value will be the upper left, just like before. This one plus the score that we get from y plus y gets us 12, and then this will continue down to 20. So using this simplified algorithm, we get some kind of measure of the similarity of these two sequences that are about 20 here. And that's perhaps more in line of what we would expect from a global alignment. This quite complex now algorithm is usually called the Needleman-Winch algorithm. And if we look at it, we solve quite a difficult computational problem by splitting it up into many small calculations which are quite easy to do, and then we look at the result. This kind of algorithm is called dynamic programming, and it has advantages and disadvantages. One disadvantage is, of course, that most of the calculations we do don't end up into the result, and that becomes an issue when we have larger sequences than these ones. We can use our matrix in order to make a traceback. We simply look at the largest number in the matrix, and we look around us, where is the second largest, and then we just follow the largest numbers up to the beginning of the sequences, and we can write this alignment out as C to C all the way to here. This means add a space into the X sequence. So we hop one nucleotide in the Y sequence and then we continue the alignment like this. So it's a quite simple, in principle quite simple problem. This is of course, as I already mentioned, computationally expensive. We make n times 3 calculations for every alignment that we do. And as I also said before, most are not used. You can use these, you use this same kind of algorithm online if you want. And there's an implementation that's called emboss needle that you can use to make this kind of alignment. It is the best algorithm, but it is also the most expensive. Let's now go back to the main motivation for doing these alignments at all. If you remember the slide with the fish and the dog and the bird, we want to compare different sequences. And if we do that with this example, we, for example, use the algorithm that we just considered, and we consider to compare these 3 sequences here. What we want to know is, these are 3 words: simulate, assimilate, and dissimilar. Which one of these are more closely related to the middle one? Is it simulate or dissimilar? We assume that there is a certain chance of mutations, a certain chance of insertions and deletions in the sequence. We just use our kind of algorithm. We calculate, we can count the number of amino acids in this case that are the same. It's 1, 2, 3, 4, 5, 6, 7. So we have 7 identities between these 2 sequences. We count these ones here. We have 1, 2, 3, 4, 5, 6, 7. So it's the same. Does that mean that these are equally similar to each other? This one to this and this one to this? Well, that depends. We need now a model of evolution, basically. How much time does it take to create a mutation that inserts these two spaces here, for instance, compared to the amount of time that it takes to change this A to an I, for instance, and possibly insert this gap here, this indel, compared to this D here, and the same in this side, T to an R, and a gap again. More importantly, if we compare transitions between amino acids, is it just as likely that we get a U to an I here as an A to an I? If we look in nature, it is not so. And this is where a tool comes in that is called scoring matrices. It's important already in the beginning to clarify that this matrix here has nothing to do with the kind of matrices we used in the Hillman-Wunsch algorithm. It's a different kind of thing. If we look at this alignment here, MGC PQR, MGL PQR, and MGI PQR, where the C stands for cysteine, one of the amino acids, L for leucine, and I for isoleucine. The question is, if we start with the middle sequence, does it take more or less time for this sequence to mutate to the 1 or to the 3 sequence? The question is, or the answer to this is that if we observe many of these sequences in nature, we will find that the transition from leucine to isoleucine is much more frequent than the transition from leucine to cysteine. Why? We can understand this in terms of chemical similarity between the amino acids. Leucine and isoleucine is more or less chemically similar. They are the same size and they have the same properties, more or less. Whereas cysteine, that has a sulfur group here, is chemically very different. So we would expect it to take more time for this mutation to occur. If we look at all the amino acids that are available to us, all the 20, we see that the ones I chose here, L and I, they are quite similar to each other in in terms of property. And cysteine is here, so it has quite different properties. But it should be said that this is an observation. We can observe this, and this is an interpretation of that observation. The person who first observed and measured this kind of differences was Margaret Dayhoff, who we can consider is the founder of bioinformatics, really. In the 1970s and 1980s, she took all the sequences in the gene bank at the time, that was much smaller than it is today, and she measured substitution rates in very similar sequences. So she took examples of sequences that were almost the same, and then 1500 or thereabout, and they were 85% similar. And then she measured what is the real frequency of certain amino acid transitions between these sequences. And she constructed this matrix that's called PAM1, point accepted mutation. And the mathematics behind this is quite complex, but we can understand these values as the likelihood for a substitution between the amino acids. If we look up our example from the previous slide, leucine and isoleucine, it's here. We have 9, there are 9 here, and if we look at leucine versus cysteine, we have 0. No. Yes, we do. We have 0. No. Yeah, we do. 0. And this means that although it's not linear, obviously, the chance for this substitution is less than the chance for the substitution of leucine towards isoleucine. So the PAM was simply made by aligning lots of sequences that were quite similar, so the alignment was not difficult. And then they actually looked at differences. Whenever there's a difference, we could use that as a so the PAM matrix was made simply by aligning these quite similar sequences, so they were very easy to align in a good way. And the difference is, whenever there was a difference, this would be taken as data for a certain transition. We look at this simple example here. This column here, A to G, and this one possibly here, would give us data for the A to G transition here, where there's the C to S here would give us data for the C to S. And then we just repeat this for all sequences and all amino acids until we have enough data to be able to construct our matrix. Nowadays the PAM matrix is not much used, but instead we use a set of matrices called Blossom, and these matrices are based on local alignments. Blossom 62 is the most used. Actually, it's usually the default for all software that compare protein sequences. And this is more robust because the sequences used for the PAM matrix were quite similar to each other, and it's possible that the substitution rates between very similar sequences is not the same as the substitution rate between any two kind of proteins. So the Blossom might be more permissive in this case. There are several different kinds of matrices we can use if we want to tweak our comparison between protein sequences. We have PAM1 to PAM250 and Blossom 100 to Blossom 30. And these matrices could be more useful for comparing sequences that are very similar, because they will penalize more mutations. On the other hand, these matrices might be more useful to measure similarity between sequences that are quite dissimilar. So what we choose depend on our use case. And as I said, normally we use as matrix in the middle here, usually the Blossom 62, and it's quite rare to change this actually in practical terms. The sequence similarity search program that we most often used is called BLAST, and it works after a slightly different principle than the algorithm that I have shown, but it's of little consequence. If we go to this link here, we can access the BLAST service that is offered by GeneBank for free, and we can type here any sequence we want, protein or a nucleotide, and we can compare the similarity between our sequence and any other sequence in the database. If we scroll a bit below on that page, we can actually see the options that we can change here regarding the matrix. As I said before, the most commonly used is Blossom 62, and we rarely change it. We can also fiddle with the gap cost of the alignment, and this gap cost is analogous to the gap penalty that we introduced in our own simple algorithm before. And although you don't actually change these options much, it's important to understand what they mean, because bioinformatics software such as this one always give you a result, and it's up to you to interpret that result to say that it's useful or informative. So this concludes what I wanted to say about bioinformatics for this course.